Okay, in 6.6 we are looking at a couple of things, independent assortment and crossing over, that are going to provide genetic diversity during meiosis. So sexual reproduction creates unique combinations of genes. Sexual reproduction creates that through independent assortment of chromosomes in meiosis. So again, when they're separating out, you're going to get different combinations to each one of the sex cells out there. And then you also have random fertilization with the gametes. So when you end up having sex, you're going to end up having only one egg, but there's going to be lots of sperm. So there's lots of different possibilities for the different gametes to get there. So that's another thing that provides variation. So unique phenotypes may also give a reproductive advantage to some organisms. So crossing over is something else that happens during meiosis that increases genetic diversity. Crossing over is the exchange of chromosome segments between homologous chromosomes. It occurs during prophase 1 of meiosis 1. I mentioned it earlier in that section's notes. It's going to result in new combination of genes. So what you see here is, like, this could be dad, and this could be mom chromosome, and then they do what is known as crossing over. It's when they form what's known as a tetrad, and then when they separate, you end up with chromosomal information with some of dad's information now with some of mom's with dad, and it would cross over in multiple spots. You wouldn't have it just do that one location. It's just showing that for this picture here. So chromosomes contain many genes. They're actually a group of linked genes. So the farther apart two genes are located on a chromosome, the more likely they are to be separated by the crossing over process. Genes that are located really close to one another are going to tend to be inherited with one another. They a lot of times are referred to being as genetically linked, or it's what's known as genetic linkage. This idea is what was used by Thomas Hunt Morgan to figure out how to map out chromosomes. So genetic linkage allows to the distance between two genes to be calculated by how often crossing over would occur. So if it happens often, then they're not very close to each other on the chromosome. If it happens rarely, then they're going to be close to one another on that chromosome. So that is the end of the notes for section 6 and the end of the notes for chapter 6.